infertile and giving them the human menopausal gonadotropin and until they start producing normal semen parameters. And the women are being referred to the gynecology clinic in SQUH. In fact, we have thalassemia major patients. We have one patient who conceived after stimulation on the first attempt, and now she's trying for a second child. And we've also collaborated in writing up some paper on fertility and birth outcome of children of women who have hemoglobinopathy. This is a paper which has been accepted for publication in Sultan Qابوس University Medical Journal, so should probably be coming in the next issues. So yes, it's a very interesting problem. Yes, they do have infertility because now they are surviving thanks to the technology, but now they're also able to conceive. And this is why now it's important for us to have assisted reproductive technology to be able to ensure healthy embryos, healthy future children for these families. Children who will not have sickle cell disease, children who will not have thalassemia. We can actually eradicate it if we want to. It's been done in other countries. Thank you.
Thank you very much, Kamal. In fact, I was looking for you, but maybe I missed seeing you. Yes, I think a theme that Dr. Marhoon also elaborated on is the increased rate of infertility is increasing. And now I have a question, maybe. In that case, since we have so much genetic diseases in Oman, that goes in the line of what we have said in your previous presentation when you said it is important that we have pre-implantation or pre-pregnancy or yes, pre-implantation genetic screening. This will help a lot in preventing these diseases and also for the marriage of the couples who are already, to reduce also the genetic disease in couples who are already married.
Yeah, in fact, I was asked to write a paper in Al-Bahith, which is like a magazine, a scientific magazine, which is produced by Sultan Qابوس University, where I address the issue of pre-implantation genetic diagnosis, the need for Oman, and more specifically for hemoglobinopathies. So yes, definitely there is a need. We're also conducting research with our medical students. The next step is about the awareness, acceptability, and the need. So we've determined that most likely this group of patients are not very well educated about their fertility options and options of having healthy children. And so we are going to address that with a research project and find the results of that to scientifically say, these are the numbers, this is the acceptance of the patient, similar to what we did with oocyte cryopreservation for oncology patients. And say, yes, we have definitely a strong justification for this. It's accepted by these patients or clients that we are treating.
Thank you very much. And I think this was presented also in a scrap meeting that you were chairing, and Dr. Muhammad Al-Marhoon presented this aspect, this important aspect. Thank you very much. Yes. Any more questions? I think we are reaching to the term of this session. Thank you very much for the speakers. Thank you very much for the audience. Thank you very much, Your Highness. You have honored us. And I think Dr. Marhoon will give us the closing remarks. I think Dr. Hani was caught in the way, so if you can give the closing remarks. Sorry, Mohammed. Sorry, I'm sorry. Okay. There is Mohammed. Mohammed, sorry, there is...
This is the end of the medical session, isn't it? Or there's another lecture? Okay, please, come in.
Sure. Okay, oh, I'm sorry, because the... مين هذا؟ My apology for the... me too, I was a bit astonished that we have three presentations, two from Oman, and then nothing from... That's right. Thank you very much. So this presentation is Professor Abbasali Khodadadi.
No, it's
No?
Khodadadi spoke
It's
yesterday. I speak. Now it's
by Dr. Masood Hushma. Okay, by Dr. Hushma. That's right. I'm sorry. My apology because Dr. Hani informed me about this modification. That's right, sir.
it's okay. I would like to thank everybody who's organized to this meeting, and I speak about what we are going to, what we do in Iran. Okay. What's the my sound is I think it's okay. What is the biotechnology health? We can do the new test for the diagnostic.
Excuse me, Dr. Masood. Maybe you can come down so that you can hear better. There are a lot of spaces here. You can come down.
Okay.
Muna, you can come down.
Well, no, I use it. We can new test for the prediction disease or disability. We can do the test for susceptibility of the disease. We can help for the test for the carrier of inheritance condition. We can do the prenatal diagnostic or new technology for the pre-implantation genetic diagnostic, which is the used in oral. But for the treatment, we can do it, enzyme replacement therapy, gene therapy, stem cell therapy, transplantation of animal to human. We can do the cloning to improve the effective of treatment and target drug therapies. If you see from we have here from pure environment and the genetics, and the meat is a multifactorial polygenic disorder as a recurrent abortion, infertility, diabetes, cancer, and I am going to speak about that. We can diagnostic from the mutation to the protein sequencing, or we can find the protein defect and go to the DNA sequencing and find the mutant phenotype. For this detection, we go from the DNA, RNA, protein, metabolism, and all of them we must go through to find a good diagnostic. But we must working in teamwork. Otherwise, everybody take, okay, this is the ball or this is the snake or something like that. But we must know the genome is the same of those things. For genetic diagnostic service, the first thing is that we need a good clinician. For those things, for the boys and girls, we can understand each other, who is the patients, and is that the autosomal recessive or what we are going to do, and we can help for understanding everywhere in this family. For the diagnosis, we can go for the human until the protein, genes, single cell, genes, and chromosomes. If you see from the moon to the Earth by telescope, is it the same as you see in the microscope one cells. The big countries or chromosome, a small of the part of the chromosome is a state, the city is the genes, and the people is a nucleotide. If somebody take a gun and kill the others, how can be detected? And if some country catch other countries, or how we can detect it? This is the country with the karyotyping and cytogenetic. We can detect all the chromosomal abnormality for 21, 18, 13, or the translocation for the different kind of the syndrome. For the state, we can do the take of the one probe and do the fluorescent in situ hybridization. Mostly it's a very quick because you don't know the growth, the culture, and for the oncology and the marker chromosome, we can use it. Mostly we use for the PGD because we have one cells and we can't have too much cells. We have just 24 hours to do it. Okay, this is the 213, 218, 221, and this is one boys. And they can use it. This one is a one boys without any dose syndrome. Most common things is coming comparative genomic hybridization. We hybrid the normal and the patient's chromosome, and we can see, okay, with the microarray, we know where is the or deletion, and now many of this microarray should be used instead of the G-banding and karyotyping, because go very fast and very details of the genetics result, and you can to 700,000 resolution instead of the 450 for the karyotyping. For the city, for the district, and the people, what we can do it, most of our diagnostic on the PCR machine, which can amplify one DNA to many after the 30 to 40 cycle of the in the machine. Yes. Okay. If you see after 30, we can see the target or band and run for the gel electrophoresis, see all bands appear or disappear. With this single test, we can do it, for example, for Duchenne muscular dystrophy, which is the many big genes, and we put many different primer and see is some band appear or disappear, and we can give the result. If you see, if the deletion of the dystrophin gene is 65%. If we couldn't find, we can go for the STR and look at, okay, this is the patient, mother, brother of the patients, and all the male family, they get a upper band of the X chromosome. But these patients, we can give the result, and this family, we give the result of all of them. They don't want married because of such many of the boys in the family died. But with this method, you can give the result. Or for the Huntington, because they have a CAG repeat, they have a anticipation from the one generation to the next generation, you can find the repeat and give the result. Okay, this is the youngest patients of Huntington I had it, and give the result to the patients for androgen receptor, for Kennedy syndrome. This is the X-linked diseases. For the same single PCR, you can do the paternity test or forensic test, which is use the different STR. Actually, we use this system for the PGD. I can tell you later about that. We have 15 different markers, STR markers, and can show, okay, which this is the father, this is the mother, and is those children belong to them or not. Or we can do it, okay, we find such a blood sample in the crime and we belong to which one. For the usual PCR, we can do it, okay, there is a band or not, but how much is it and how many copy number we have. For the viruses, we use the real-time PCR, we put it the different fluorescent system and give the result, okay, this many virus copy number can lead to the disease or not. Mostly we use the sequencing method to see all the nucleotide in the patient's genes, target genes. For the metabolic disorder as a Niemann-Pick, Gaucher, because we don't know the mutation in our country, we go sequence all the genes. This is the sum of the result of the lysosomal storage disease. Another technique we use the southern blot analysis for the dose they have it because of the PCR can't do the CGG repeat on it for the Fragile X, for the myotonic dystrophy, for the facioscapulohumeral dystrophy, it's a neuromuscular disorder, which is can use it the southern blot analysis. Because the name of the inventor is Southern, and another for the DNA and RNA, they do the northern blot and western blot. Western blot is a very good analysis for those many genes has it in the cause of the disease as a immunodeficiency, such a SCID. If I want to do it all of them together, it's very costly. I do it the one western blot, find which of those is a deficiency, and then go for the genetic analysis. For the CGD, it's because 8 genes involved for the viscodaldrich, glycely, or neutropenia. I use this technique first and then go to the sequencing. MLPA is a very good system for the copy number of the disease. Even if you failed amniotic for the do the karyotyping, you can do it QF-PCR, do it see the X, 21, 20, 18, and 13, and Y chromosome is normal or not. I use it for the Charcot-Marie-Tooth, which is the PMP22. It's the most 70% belong to duplication, and I can find the copy number of this, and this is the result of the Charcot-Marie-Tooth. Reverse dot blot is a very easy for the beta thalassemia, very comfort. In one day you can give the result to the patients, and there is a company sell these things, and I use it mostly for the thalassemia. Microarray technology is a very good. Now they are going to use it for the multi-genetic and multi-mutations as a hearing loss, Leber congenital amaurosis, Bardet-Biedl, and you can find many different diseases in the same chip and check for the family. From the DNA sequencing, SSCP, marker, MLPA go more and more for using of that. One genes, one disease, we use those technology, but for the all genes and all disease mostly go for whole genome sequencing, which is the next generation sequencing array. Comparative genome hybridization is a good. This is the medical. We have more than 400 tested for immunodeficiency, neuromuscular, metabolic, and multi-genetic disorder. My suggestion for collaboration to establish diagnostic service center between two countries for single and multi-genic disorder, find the new genes for the rare disease by next generation sequencing. We can do the one referral lab, some lab sent to this one, and we find a service lab in the different part of the Oman and Iran and put it together. PGD is we can take one cells of the IVF or ICSI and examine for the diseases. Those diseases can go for the PGD by now and be more and more tests. The second suggestion for collaboration, establish diagnostic service center between two countries for PGD, because many of the people don't like the abortion. We can say, okay, this cell blastomer is okay or not, and use it for the, put it back for the delivery. In multifactorial disorder, cancer, multiple sclerosis, recurrent abortion, infertility, diabetes, Alzheimer, cardiovascular, and autism is a very difficult but very common disorder. I suggest we can working with multifactorial for the prediction, prevention, diagnostic, prognostic, personal medicine, and therapy. Give the service, do the kit, research, and a student for that. For example, for cancer, we can take a blood and with the cancer stem cell see, okay, this patient have a cancer or not. Another test for which kind of cancer they have. For diagnostic, prevention, prediction, prognostic, personal medicine, which is very important in the cancer, and therapy, for cell therapy and energy therapy, we can do it with the different kind of the cancer. Suggestion, collaboration for established diagnostic service for prediction, prognostic, prevention, diagnostic, and personal medicine and therapy for multifactorial disorders, especially in the cancer area. Metabolic disorder, because it's a very, we have a 4,000 to 5,000 metabolic disorder, 400 to 500 enzyme deficiency with MSMS and GCMS. We can define and find the enzyme deficiency. We must working with the metabolic, enzyme mutation, and genetic counseling all together. Some of them for the micromolecular, they have a diet. If we find a very quick the patients, we just change the milk of the patients, we can cure the patients. For this macromolecular as a lysosomal mitochondrial, we can do the enzyme replacement therapy. My suggestion is that we do the metabolic disorder, newborn screening, not for 2 to 5 disease. We can do it to 28 disease together, enzyme assay, gene, genetic diagnostic, and we can help with the biotechnology, do the food production for the micromolecular as they have a diet, and cell therapy for macromolecular and enzyme replacement therapy. My suggestion that we establish center service for between 2 countries for metabolic disease for diagnostic and treatment. Suggestion 5 for food production, we can do it with the biotechnology in our centers, can help here in the Sultan Qabus University to find the production by biotechnology, food production for the metabolic disorder, enzyme replacement therapy, stem cell therapy, and herbal medicine, which is the very important in this issue. The last, my suggestion for collaboration is a medical genetic and genetic counseling program for medical students, because the first thing is we must have a very good clinician and know about the genetics, and MSc and PhD students education in medical and biomedical genetics. Thank you very much for your attention.
Thank you very much, Dr. Masoud, for this very comprehensive and in-depth review about the genetic diseases and the scope of cooperation. Can you give us an idea about Hushman Genetic Laboratory?
Yes, I have 2 laboratories. One of them is in the National Institute for Genetic Engineering, and another is my own laboratory, is a Hushman Genetic Laboratory, is my own. But because we have many patients, don't have too much money or have a problem, we do it in the governmental section, and another want to come in the afternoon, come to my own laboratory.
Thank you.
Dr. Masoud, thank you very much. I'm just putting a simplistic question. It sounds that by genetics you can diagnose a big number of diseases. So do you think it is better to start with genetic work up from the beginning as a basic evaluation of the patient?
I think the first thing is the doctor, see the patients, then because the genetic service is expensive. Some of them you can diagnose with one MRI. Some of them with lactate and private you can do your diagnosis. Something with the pathology you can give the diagnostic. But if they want for the next generation, because many of the enzyme is not expressed under the fetus and under the pregnancy, and therefore you must have a genetic test, because in the 10 of 2 weeks of the pregnancy we can do the diagnostic for the patients. And therefore for the diagnostic mostly we go for the cheaper way, but if they want planning for to have a more child, or if or diagnostic help the patients for treatment, we do the genetic test.
Yeah, you are right, absolutely. They are expensive, but sometimes we do a lot of tests, and the diagnosis takes a long time until we reach to a diagnosis. In my feeling, if indicated in special societies or families, or when you think there is a possibility of genetics, why not to start with the genetic evaluation?
Yes, thank you. Yes, please.
I have a question regarding PGD. Do you have any data about your success rate of pregnancy after doing PGD?
PGD is very important. Every prenatal or PGD, you must know the genetic exact in the family, first of all. 2, you must have a good instrument and service, because if you go for you can go for the sequencing for blood, but you can't go for the one cells, because they have 2 copies. If in each reason one of the copies is not amplified for the PCR, you go, which called allelic dropout, you go for the wrong diagnostic. You must go for 3 or 4 STR inside and near the genes and do the diagnostic. And the rate of the PGD, the same of the rate of the IVF or ICSI, is 25 to 35%, not more. If somebody said, okay, if coming the PGD and they be pregnant and everything's okay. It's not. But everywhere, when you do the PGD, you must give the permission from the patients. You must do the prenatal diagnostic as well. Otherwise, it's something wrong and you can't give the answer.
How much it costs in Iran?
The cost of the PGD is calculated between 3,500 to 5,000, depend on the disease, because some disease, as thalassemia, everything you know about that, you know about the STR, you know the gene is very small. But if you want to go to the other disease, which is you must set up everything for the single patient, the cost be more.
Yes, Dr. Maha.
Right, thank you. Okay. Thank you for a very interesting presentation, quite informative. I'd like to ask, have you started with fetal DNA prenatal diagnosis from maternal serum? You may be aware that it's already being used in clinical capacity. North America has it, and United Kingdom. People from this part of the world are sending the tests there to get the results. How about Iran?
If you want to
Excuse
go
me, Dr. Masoud,
to
can you
see
repeat the
at
question?
the patients.
Excuse me, Dr. Masoud, can you
Yes.
repeat the question?
The question is uses the fetal cells in the serum of the mothers. If we take a blood sample from mother, the pregnant mother, can we get the diagnostic of the cells is running in the mother's blood or not? This is the very good question. Actually, if you want to see the fetus is a male or female, is okay. If you want to see the Down syndrome, sometimes. But if you want to go to because you must have at least 1 liter to take many fetal cells to go with the diagnostic with a special disease. But for in Italy, they do it such a machine with the half liter of the blood of the mother can separate it and then go for the diagnostic. But it's not coming for the commercial yet. You can detect at your fetus is a male or not. Some of the laboratory do it with the Down syndrome and like that, but they must repeat it with if they have actually legal medicine don't give you the permission for abortion if you just say it, I do it with fetal cells.
My question is actually, is it available? Is the test available?
Yes,
in your
available.
laboratories in Iran? And it's usually now used for trisomy. It's not.
No, it's available in the 10 to 14 weeks of the pregnancy. We can use it. But if they have such a special disease, I am not recommended for that.
What is your success rate? Like what's the success rate of picking up a trisomy?
It's about 70 to 80. We complete this one with the quadrat test or triple test. We do it with alpha fetoprotein and like that. If all of them together be sure that there is a risk for the Down syndrome, we do it the karyotyping for amniocentesis. In the 10 to 14, we do the 2 tests. 1 test for the fetal cell, another for quadrat test, which is the alpha fetoprotein, beta ICG, and like that we measure it.
Any questions? Anna, you seem to have a question.
Can I have a question?
Yes. Okay. So, Dr. Muhammad, what do you think of it?
Yes, Dr. Masoud,
Yes.
we do know that pre-implantation genetics has a risk of embryo loss. So how do you reduce this, or how do you go about it?
Embryo loss, actually, every woman can get pregnant, and before the next period, they get lost the child. The rate of everything is 25 to 35. Is it the same? PGD is because we take 1 cell, it must be such a good assistant or good technician, which is the one, actually we want 2 cells because those must confirm each other. For the 10 of the eggs, which is the examine, actually 4 of them is not used after that. And therefore, you have just a 6 embryo. Of the 6 embryo, we put it 2 or 3 in the each time because we want to go more success. And therefore, many of the IVF child or the PGD child is twins or more. And therefore, the rate of the success is 25 to 30.
So are you saying it is a must to be done in all the assisted reproductive techniques and labs?
Yes.
And if so, what about the added expense to this? Because the IVF-X is already expensive, and you are adding another cost by this pre-implantation genetics.
No, this is, I said at $3,500 to $5,000 for all the package, not the additional for the IVF or like that.
Thank you.
Dr. Masoud, there was, I think, in the New England Journal of Medicine, when they compared the WHO, the Genome Project, and compared it with the whole genome sequencing, one of the comments was said that, yes, in the WHO project, they were looking for common diseases, and then they were looking for the mutations in these areas. While in the whole genome sequencing, we look what is it, and then we see the diseases. But still, in their comment, they said in what we call the whole genome sequencing, we don't sequence the whole genome.
No,
we sequence certain areas.
Yes.
So still there is possibility that we miss some of the diseases.
Yes, of course. This is called wrong at the whole genome sequencing. This is we do the exome sequencing because all the exomes are not too much compared to all of our genes. We don't go for the intron or some region for replication of the genes and like that. We go for the exome genes, and the analysis is very important because the analysis of that, I met some of the big guys, I can't remember. DNA پیدا کرد کی بود؟ Oh, Watson and Greek, one of them is died, another I met him in Canada. I said, because of my son get the schizophrenia, I do the whole genome sequencing. And when I saw the result, I be mad because it's too much result, too much polymorphism, too much things, and it's not very easy. One of the people, they do all the gene sequencing, not the exome sequencing, is Watson at because it's bioinformatics is more important in this issue. You find a homozygote mutation, change the amino acid, but it's a polymorphism. You must, after they find it, go for the family, sequence that part for all the healthy in the family. If they have a homozygote in this part, is a polymorphism. If not, go for at least 100 healthy people, show that this is not, this is causative or not.
Thank you very much. Well, I give now the microphone to my co-chair.
Thank you very much. I think we have finished our time for the medical session, and we'll leave the closing remarks to Dr. Hani afternoon. And so, please, the next session to be commenced now. Thank you.
The next speaker is Professor Saif Al-Bahri from College of Science, Biology Department. His presentation will be about the current biotechnology research and applications at Sultan Qابوس University.
Okay, thank you very much for this. What I'm going to give, in fact, is a showcase of what we are doing. And the reason is that to find out where are the ways, or what are the ways, or which areas that we can collaborate. So the first thing that I will start is actually, because of the medical people are here also, is the major problem that we are facing about antibiotic resistance. And actually, whatever we give in hospital, it will go to the environment, and actually it will end up in the food chain and come back to human being. That's the thing that is actually is coming. In fact, this paper we have sent it for as a review for the last, probably from 1985 to now, the experience that I have in this area. And it's very interesting that just in April, about 2 weeks ago, April 2014, is the first time WHO is actually producing like this result, showing how serious it is about the antibiotic resistance. And what we're going to face is a very, very bad situation, it looks. And sadly enough, that if you look about Oman, there is almost very little known about this. So my studies were actually based on my background, where I started actually working in the Ministry of Health before, then joining the Sultan Qابوس University. So I have that experience, what is happening there, especially when I was in the preventive medicine. There where I worked with clinical samples as well as water and food samples. And of course, I used that to see how it is. So usually you find there is a lot of tetracycline in particular is used in growing chicken and so on like that, fowl in general. And we find that that tetracycline, it goes to the organs of the chicken and we eat it. And we find actually these are the contents of the tetracycline in different tissue. Of course, we published several papers in this area, but the very, very worrisome thing is that you can see that only in tetracycline, of course, we did a lot. In some cases, we isolated the microbes. At least we tested 15 antibiotics, but 14 antibiotics, the microbes were resistant to this. So, and we find that, in fact, one of the studies that we did, we started to concentrate tetracycline and picoline because environmentally it was the highest. That's the reason. And we find that for the first time, we did not find any paper that in tetracycline, the microbes, the E. coli here, which is our normal flora usually, has 4 TET genes combination resistance is very worrisome. And in fact, we checked it from chicken, from human beings, and from the treated sewage effluent, where we think that sewage effluent is a meeting point where the microbes are meeting and exchange the genes and so on. The other thing that we did is, in fact, how there is a lot of food poisoning that is happening here, and we find out that this food poisoning is, in fact, with the antibiotic resistance microbes. So we did these studies here with the spoilage food, and this is here the actual study, electron micrograph, talking about the penetration of the microbes into the chicken eggs, how it is. And we did this study anyway. So in that case, sorry, it's a mistake of spelling there, but how we can use antibiotic-resistant bacteria as bioindicators of pollution. This is what we did. So we checked the sewage effluent. Usually this is treated sewage effluent. And here we look at the fisheries, farmlands, and so on like that. And we published several papers related to this issue, but we used STU as it is a closed system where our sewage is released into our environment here, in soil and so on. And it is very interesting that we took, this is our STP here. There is a very good chlorination. You'll find 0 microbes are growing there. But as you move away, this is nearby the College of Commerce there. We collected a point there, and we find there is a regrowth of the microbes. But the ones that are here you find 0, but the ones that are there, they are fully antibiotic-resistant microbes. So this is a very good indication that from sewage, which is a meeting point, it goes to the environment. And that is very serious, and that is where actually in Oman ourselves, live outside Oman, but in Oman ourselves, we have to collaborate to find out and solve this particular problem because of the seriousness according to the WHO report. And you can find that we check this in fish, we find that in fish, and we find it also in turtles, that there are also antibiotic resistance. How the turtles got antibiotic resistance, or fish? In this particular one, we did in Dorset, we can see there is a sewage treatment plant. In a very similar way, it is actually with chlorination, no microbes, and suddenly when it dumps to the sea, we find actually even